Hello everyone, welcome to the Concept and Fact of Life Sciences. Today in this video, we are going to discuss about the cell organelle, that is lysosome. As we all know that the lysosome, like endoplasmic reticulum, it is a single membrane bound cell organ. These lysosomes, these are also known as the suicidal bags of the cell. Why these are called as the suicidal bags of the cell? Because these lysosomes, they contains 50 different type of hydrolytic enzymes which can carry out the digestion of any material. And if the lysosome they burst accidentally in a huge quantity, then these acid hydrolases or the hydrolytic enzymes they comes into the cell cytoplasm where they can carry out the autophagy. That's why these lysosomes they are called as the suicidal bags of the cell. Now these hydrolytic enzymes which remain present within the lysosomes, they are called as the acid hydrolases. They are made up of two words, that is acid and the hydrolases. Acid means they require acidic environment for their functioning, and hydrolases means they require water molecule to carry out the breakdown of other molecules. So first thing, there should be a presence of acidic environment for the functioning of these hydrolytic enzyme or the acid hydrolases, and they will carry out the breakdown of molecules by using the water molecules. So these are the hydrolases. Now within the lysosome, suppose this is the structure of lysosome, there is a presence of a single membrane. The pH of lysosome, it is around 4.5 to 5, because this pH is essential for the functioning of enzyme within the lysosome, or we can say for the appropriate functioning of acid hydrolases within the lysosome. Now how this pH it is maintained? It is maintained with the help of some special type of pumps which are called as V-type ATPases. जैसे कि अपन ने पहले बात की थी जब अपन ने मेम्ब्रेन ट्रांसपोर्टेशन पढ़ा था, there is a three types of ATPases or pumps involved in the process of active transportation, which are P-type pumps, V-type pumps, and the F-type pumps. So there is a presence of V-type pumps. These were firstly discovered on the vacuolar membrane. That's why these are known as V-type pumps. They also remain present onto the membrane of lysosome. Now, on the membrane of lysosome, there is a presence of F-type ATPases. These ATPases, they utilizes the energy in the form of ATP and will carry out the transportation of H plus ion against their concentrational gradient. Means they will carry out the transportation of H plus ion from, they will carry out the transportation of H plus ion from their lower concentration to the higher concentration. So these are involved in the process of active transportation. Now, due to the functioning of this V-type ATPases, the H plus ion concentration it remains high within the lysosome, that is responsible for maintaining the pH of compartment around 4.5 to 5, which is essential for the functioning of enzymes which are known as acid hydrolases. Now, if these acid hydrolases they comes into the cell cytoplasm, the pH of cell cytoplasm it is around 7, that is slight alkalinic. These acid hydrolases they are not able to function over here, but the autophagy can occur. इसीलिए अपन ने पहले बात की थी कि if the lysosome they burst in a larger quantity than these acid hydrolases, because the environment or the cell cytoplasm it itself become acidic if the lysosome they burst in a large amount or large quantity. In that particular case, they can perform their function within the cytoplasm and can carry out the autophagy. If one or two lysosomes they burst, they are not going to function within the cell cytoplasm, so autophagy will not occur. So this is all about the structure of lysosome. If we talk about the functions of lysosome, the first and the most important function of lysosome is the digestion. Now the digestion, it is of two types. One is known as intracellular type of digestion, while the other is known as the extracellular digestion. The lysosomes, they are mainly involved in the process of intracellular digestion. Now, how this intracellular digestion it occurs? In this intracellular digestion, the cell, it first recognizes the food particle. Suppose it is any food particle, and it is the phagocytotic cell. So the cell, it will first recognizes this food particle, then this food particle, it is engulfed by this phagocytotic cell. So there is engulfment of this food particle. This vesicle, food vesicle, it gets detached from the outer membrane, comes into the cytoplasm, where there is a presence of lysosome containing the hydrolytic enzymes. The food vesicle fuses with the lysosome that lead to the formation of a phagolysosome, and the digestion occurs within this phagolysosome. जैसा अपन बात कर सकते हैं, food particle is first recognized, then the food particle, it is engulfed by the cell. Digestion occurs inside the cell within the phagolysosome. This is the way of digestion in some lower animals. For example, the amoeba. It recognizes the particle, then there is an engulfment of particle, and the digestion occurs within the cell. That is the intracellular type of digestion. Some phagocytotic cell, or we can say this type of intracellular digestion also occurs in the higher animal like human. There is a presence of some phagocytotic cell in the form of neutrophils and the macrophages that also attacks, or we can say that also recognizes and engulfs the foreign particle like the bacterial cell, and the digestion occurs within these cells. So in this process of intracellular digestion, the lysosomal enzymes plays an important role. Another type of digestion, it is known as the extracellular type of digestion. In case of the extracellular digestion, the cell secretes certain enzymes. For example, the pepsin and the trypsin, which carries out the digestion of the food particle. And after the digestion, the food particle, or we can say the digested particles, they will get absorbed by the cell. So it is a predominant way of digestion in the higher animal. Some fungus, they also have extracellular digestion, where some saprophytic fungi, they releases certain enzymes in the surrounding, which will carry out the digestion of we can say food particles. Then the digested material, it is absorbed by the fungal cell. So there is a two type of digestion, and the lysosomes, they plays an important role in the intracellular type of digestion. Next type of function of lysosome, it is the autophagy. Autophagy, जैसा कि अपन ने पहले बात की थी, if there is a bursting of lysosome in a large number, then it will create the problem for a cell in the form of autophagy, or where it can digest the cellular compartments. Okay, but if it occurs in a controlled manner, the autophagy, if it occurs in a controlled manner, it is useful for the cell. For example, this autophagy, it is involved in the removal of damaged organelle, which is, or we can say, for example, the mitophagy. Mitophagy, जैसा कि अपन ने माइटोकॉन्ड्रिया के अंदर पढ़ा था, there is a selective removal of damaged mitochondria from the cell. And in this process of mitophagy, the lysosomes, they plays an important role. Next thing, these lysosomes, they are also involved in the process of nutrient recycling. Means if there is some misfolded protein, if there is some unwanted nucleic acid are present over here, then these lysosomes, they have ability to convert these nucleic acid into the nucleotides, or proteins or misfolded enzymes into the amino acid, which can be utilized in the further cycles of translation. So they are involved in the process of nutrient recycling. They are involved in the process of protein quality check. Now the degradation of protein, it occurs at two places. One in the lysosome, other in the proteasome. The degradation which occurs within the proteasome. It is a ubiquitin mediated degradation where there is an attachment of polyubiquitin to the protein, and the degradation of protein occurs within the proteasome, which is a part of cell cytoplasm, जैसा अपन ने पहले बात की थी. Now, if we can say there is an attachment of monoubiquitin, or if there is a ubiquitin independent type of protein degradation, then the degradation it occurs within the lysosome. So lysosome, they are generally involved in the ubiquitin independent degradation of proteins. So if there is a misfolded protein, if there is a misfolded protein, then it will be sent to the lysosome for the degradation. Next is the process of cellular homeostasis. So by doing all these things, means by removing the selectively removing the damaged cell organelle, by carrying out the process of nutrient recycling, with the help they also help in the process of protein quality control by the degradation of non-functional protein or misfolded protein. They will carry out, or they are involved in the process of cellular homeostasis. So this is the second function of lysosomes. Next is the programmed cell death. Now the cell death, they are generally are of two types. One is known as necrosis and other is known as the apoptosis, which is also called as the programmed cell death or the PCD. Now these lysosomes, they are involved in both of these processes, apoptosis and the necrosis. Likewise, within the lysosomes, there is a presence of procathepsin. Okay, suppose this is the structure of lysosome, and if there is a lysosomal membrane permeabilization, LMP, if the LMP it occurs, then there is a leaking of content from the lysosome. During this process, the procathepsin, they will convert into the cathepsins. After this, or we can say after the conversion of procathepsin into the cathepsin, these cathepsin, they will further activate the cysteine aspartate proteases, which are known as caspases. And these caspases, they are involved, or these are responsible for the process which is known as the apoptosis or the programmed cell death. दो अलग अलग तरीके के पाथवे होते हैं, उनके बारे में अपन आगे बात करेंगे. अभी सिर्फ इतना समझना है कि lysosome, they are also involved in the process of programmed cell death and the necrosis. If this leakage it occurs in huge quantity, under that particular condition, we can say the cell will undergo the process of autophagy, and there is a process that is called as necrosis, which is called as the accidental cell death. So we can say that the lysosomes, they are involved in both of the processes, that is the programmed cell death and the necrosis. If there is a leakage which is under control or under regulated, or we can say it occurs in a smaller amount, under that particular condition, there is a conversion of procathepsin into the cathepsin. There is an activation of caspase which lead to the apoptosis. If leakage occur in a huge quantity, then the cell will undergo the necrosis. Next function, it is the cell signaling. Now, this is the structure of a lysosome. Okay. Now, if there is a presence, or we can say if the cell is in the nutrient rich state, there is a sensing of amino acid, that is leucine and arginine, within the lysosome. After sensing these amino acid, particularly the leucine and the arginine, there is a activation of RAG protein. This RAG, it recruits the GTP and will convert into the RAG-GTPase. That is the activated form of RAG protein, and the attachment of GTP, it only takes place after sensing the amino acid, that is leucine and arginine. After sensing these amino acids, the RAG, it recruits the GTP and it will convert into the activated form, which is known as RAG-GTPase. Now this activated RAG, that is the RAG-GTPase, it recruits another protein to the surface of lysosome, and this is known as mTORC1, which is the mechanical targeting of rifampicin complex 1. The recruitment of this mTORC1 onto the membrane of lysosome, it will carry out very many functions. It is involved in the process of lipid biosynthesis. It activates the ribosome-mediated protein synthesization, and it also inhibits the process of autophagy. So, in this way, we can say that it is involved in the process of cell growth and regulation. So, the lysosomal signaling, it is involved in the process of cell growth and regulation. Now, next function, it is the bone resorption. Now, what is bone resorption? Likewise, there are cells which are known as osteoclasts, and there is a cell which are known as osteoblast. So there are two types of cell which maintains the calcium homeostasis. So we are talking about the osteoclast. Suppose if there is a deficiency of calcium within the cell, then these osteoclasts, which contains the lysosomal enzyme, it degrades the bones and will carry out the mobilization of calcium from bones to the cell, because we are under the deficient condition of calcium. So osteoclast, they contains the lysosome and the lysosomal enzyme as well as the pH, that is the acidic pH. It is responsible for the degradation of old bone matrix, that will carry out the mobilization of calcium to the cell, and which will meet the deficiency of calcium within the cell. So they are involved in the process of calcium homeostasis also. Next function, it is the lysosomal storage diseases. Now the diseases which are due to the non-functioning of lysosomal enzymes are called as the lysosomal storage disease. Because these enzymes which remain present within the lysosome, they are called as hydrolases, acid hydrolases, which will carry out the breakdown of larger macromolecules. So if there is an absence of these hydrolases, the we can say the digestion will not occur, and there is a storage disease which are known as lysosomal storage disease. Now these lysosomal storage disease, these are inherited metabolic disorder, and most of them, they are the autosomal recessive. So if we talk about the first disease, which is known as Tay-Sachs syndrome, there is a deficiency of lysosomal enzyme which is known as hexaminidase A, and due to the deficiency of this particular enzyme, there is an accumulation of GM2 gangliosides. So if there is a deficiency of this enzyme, there is an accumulation of GM2 ganglioside, and the disease is known as Tay-Sachs syndrome. Second is the Gaucher disease. In the Gaucher disease, there is a non-functioning or absence of glucocerebrosidases. Now these glucocerebrosidases, they will carry out the breakdown of glucocerebrosides, and under this their deficiency or in their non-functioning condition, there is an accumulation of glucocerebrosides which lead to the disease which is known as Gaucher disease. Next is the Niemann-Pick syndrome, where there is an absence of sphingomyelinase, and if there is an absence of sphingomyelinase, there is an accumulation of sphingomyelin that lead to the disease which is known as Niemann-Pick syndrome. Pick disease, we can say. Next is the Pompe disease. Pompe disease, it is due to the non-functioning of glucosidase, and if the glucosidase enzyme, it remain absent, then there is a storage of glycogen. germ which is responsible for Pompe disease. So this is all about the structure and the function of lysosome. Thank you.